So Mendel crossed plants from the F2 generation, and he found that there was one trait that was always dominant. Now put on your Mendel thinking caps, go back to the 1800s, and try to draw conclusions based from these observations like Mendel did. Okay, you don't know as much as we do now about alleles, etc. So hopefully you concluded that parents' traits do not blend together in their offspring, and that one version of the trait is lost during reproduction. Now, this is not necessarily true, but this is what you can conclude from the experiments that Mendel carried out and from knowing what Mendel knew at that time. Okay, let's move on. So Mendel concluded that the traits do not blend together in the offspring of two parents. The blending hypothesis can be rejected. The reason that everyone thought blending was right was because they never looked at individual traits, which means they looked at the overall organism and found that overall there seemed to be a blending. Okay. Now look what happens when I breed the two F2 generation tall plants together. So when you breed the two F2 tall plants together, the F3 generation has a distribution of tall and short plants, and this is the order. Okay. With these new data, what should I conclude? Note that there may be more than one good answer to this question. So again, pause for a minute and think. What can you conclude from observing what you just did over here? And from this experiment, you can conclude that this is additional evidence that traits do not blend together in the offspring of two parents. And we've just disproven one thing that we said might be true earlier, and we say that each of the different versions of that trait maintain a discrete identity in the offspring, and therefore they appear in the F3 generation. Okay, so I concluded that the factors that control a trait maintain a discrete identity when passed from parent to offspring. This became known as my law of segregation. And you know this is true because when you studied meiosis, you saw that a sperm or egg carries only one allele. In Gregor's terms, trait. Only one trait for each inherited character because allele pairs or trait pairs separate, that means segregate, from each other during the production of gametes. Until today, we know this to be the law of segregation. Next question. In humans, the allele for glaucoma is dominant over the allele for normal eyes. Suppose a man with glaucoma marries a woman with normal eyes and they have children. If this trait is inherited like height in pea plants, which of the following would be true? Again, take a minute, pause, and write down the answer you think that might be true for this situation. Okay. So, did you think of C? Some of their children may have glaucoma, and if the children with glaucoma reproduce with someone who has glaucoma, most of those people will have glaucoma, but some will have normal eyes. Back to Mendel. But I noticed something strange. The dominant traits were always more common than the recessive traits. So, for example, when he carried out the following experiments, where he crossed round versus wrinkled, yellow versus green, full versus constricted, etc., etc., he always found that the offspring were present in this ratio. Okay? One was always more dominant than the other, and that is called the dominant trait, and the one that appears in fewer numbers is the recessive trait. And they were in almost the same ratio. Remember we calculated ratios in the lab? This is how he calculated it, the number of dominants divided by the number of recessives. And he always found that the dominant traits appeared 3 times more often than the recessive trait. In other words, 75% of offspring show dominant traits, 25% of offspring show recessive traits. How could you explain this? puttered Gregor. My mathematical training in the university came to my aid. My physics professor, Herr Doppler, always encouraged us to think mathematically. I asked myself, could I explain this 3:1 ratio simply? Yes, yes, and again, yes. So let's see how we can explain that 3:1 ratio in offspring, 3 dominant versus 1 recessive. Suppose that the traits were factors like particles. We could list them this way. The dominant factor, represented by capital T, stands for the tall plant. The recessive factor, represented by small t, stands for short plant. Imagine that a purebred tall plant would have 2 capital T's and a purebred short plant would have 2 small t's. A purebred would always be homozygous. Then, when I breed them, a tall versus a short, the tall bearing 2 tall alleles, the short bearing 2 short alleles, because these are homozygous traits. In a true breed, they will always be homozygous. If they would pass on one of their traits via the pollen or the egg, that is the ova, then what? I have it, father, I have it. The answer would be 1 capital T and 1 small t. But father, that would mean that the tall hybrids would be carrying a factor for shortness. Exactly. But you cannot see the shortness. It is hidden. And what would happen when 2 hybrids were bred? That means one organism carrying the tall T and the short T versus another organism carrying the tall T and the short T. I know, father, there would be 1 pure tall plant, 2 hybrid plants, and 1 short plant. Exactly. Mendel analyzes the data. Okay, so this is the same thing that we talked about in the earlier slide, just a pictorial representation of the dialogue that Mendel has just gone through with his students. Okay. So let's work it out. One parent has the capital T and 1 small t allele. Another parent also has a capital T and a small t allele. When they segregate, each gamete will have either the capital T or the small t, but not both because of the law of segregation. On the other side, the 2 gametes that are formed here will have either the capital T or the small t, not both. Again, the law of segregation of alleles. Now, when a gamete from one parent fuses with a gamete from another parent, you could have various combinations of this fusing. For example, if the capital T fuses with this capital T, then the offspring will be 2 capital T's. If the small t fuses with this small t, then the offspring will be 2 small t's. If the capital T fuses with this small t, then the offspring will have 1 capital, 1 small t. If the small t fuses with the capital T, then the offspring will be 1 small t, 1 capital T. So the genotypic ratio is all different. The genotypes are not the same. There's no ratio here. The genotypes here are all different, as you can see. Small t, capital T, small t, 2 capital T's and 2 small t's. But what about the phenotype? The phenotype here shows you that this plant is tall, this plant is tall, this plant is tall, but this plant has no tallness genes, so this plant is short, which means there are 3 tall plants to 1 short plant. Therefore, the 3:1 ratio representing the 3 dominant phenotype bearing 3 dominant capital T bearing organisms with 1 recessive bearing organism. Okay, think about this. Now look at the glaucoma question again. What information would be most helpful to you in predicting with more certainty whether the offspring of these parents will have glaucoma or normal eyes? So the situation was a man with glaucoma, which is a dominant trait, marries a woman with normal eyes, and having normal eyes is a recessive trait, and they have children. Okay, so hopefully you thought of C and D. you need to know whether the man is pure for glaucoma. So is he homozygous with the capital G, if you say G stands for glaucoma, or whether the woman is pure for normal eyes. In that case, small g, homozygous for normal. You can derive that, come to that answer mathematically, like Gregor Mendel did, simply by using a Punnett square. Okay, what does a Punnett square involve? It involves drawing a square. You see over here. Okay, then you divide that square into 4 small squares. You write down on one side of the square the alleles that are inherited from one parent. For example, over here we've represented the alleles that come from the eggs or the ova, okay, which is a capital T, small t. We're going back to the tall plant and the short plant experiment. And on the other side, you write down the alleles that come from the pollen, which again is a capital T and a small t. So we're talking about a heterozygous plant. You can predict the genotypes of the offspring by carrying down one of these alleles into each of the squares, one from up there, one from here. So what really you're showing you is the 4 possible outcomes of a cross between a heterozygous TT and a heterozygous TT. So if you pull down one allele from each of those gametes, like so, then you can see that the offspring have these 4 combinations of alleles. This is what we refer to as a homozygous dominant trait. This is a heterozygous trait, in which case the capital T will be dominant. This is a homozygous recessive trait, and this is a heterozygous trait, of which the capital T is representing the dominant trait. Okay, so what we've covered so far are the following terms and principles. We talked about what is a dominant trait, what's a recessive trait, a monohybrid cross. We began that in slide 19, a cross involving one trait. We haven't talked about a dihybrid cross, but that is something similar to a monohybrid cross, but instead of following only one trait, for example, only looking at tallness or only looking at peak color, we follow an experiment, or we carry out an experiment following 2 traits. That means looking at tallness and peak color at the same time. And we'll talk about that next week. We talked about phenotype, genotype, and we are going to talk about the particulate theory of inheritance. That means you need 2 particles of factors to determine a trait. Okay. What else have we covered? We've covered what are alleles, we've covered homozygous dominant and recessive traits, heterozygous traits, and a Punnett square. So we'll stop here for now and then continue in our next video.